Okay, ladies and gentlemen, let's get started. Let's just take a look at the syllabus. Go ahead and grab your syllabus and let's make sure we all are on the same page. See where we know where we're going. Today it's Monday, what is today? June 8th. Wow. And if you look at the syllabus, we have an exam next Tuesday. Okay. That exam is going to cover what? Chapters 3, 4, and 5. 3, we've been on for a little while. That was cells and organelles, and I'm going to finish up the transcription translation story with you here. And then 4 is histology. You've already had a lab on histology. You're all going to be quizzed on that histology this week in lab. So that's not a brand new story. I will re-lecture on highlights of that, and while I'm doing that, you'll be picking up some new details and hopefully connecting some dots. And then finally, chapter 5 is on skin, the integumentary system, and it'll be our first system that we'll be looking at. And in fact, it's very straightforward. So I'll be finishing up 3 today, getting into 4. Finishing up the last little bit of 4 on Wednesday, and definitely getting through 5. Wherever I get to the end of on Wednesday will be the place where we stop and what will be included on the exam. So I'll make any adjustments if necessary. That vocabulary on exam 2 is going to be what? 21 through... Who has their syllabus out? 21 through 40. And last time I didn't do any vocabulary with you. So that means I need to do 10 slides today and 10 slides with you on Wednesday, just to be honest, to go through those with you. So let me start off by going through vocabulary. 21 through 30 today, 31 through 40 with you on Wednesday. Hopefully you've already taken a look at those. And that's the vocabulary that will be on exam 2. Now it is non-cumulative. So the vocabulary will just be 21 through 40. It will not be going back to 1 through 20. Hopefully you remember some of that, but I won't be quizzing you directly on that. So are there any questions on just logistics, housekeeping things related to what we've done so far? I'll answer any questions in a few minutes, but just housekeeping right now and on what's exam number 2 next week. If you are in the hybrid section and you're listening in, then you'll want to schedule with the testing center your exam next Monday. Keep in mind, the more limited your availability is, the sooner you need to schedule that appointment, because they do fill up sometimes. So make sure if you have a very limited availability next Monday that you have called in. They've already got slots ready for you for next Monday for the exam. Okay, so no housekeeping things. Let's take a look. Vocabulary. Is it through 40 or 41? Through 40? So we're going to be going cutie through histero. Okay, this next exam, cutie through histero. So you can go ahead and make up those slides now. So cutie, like cuticle, right? That little piece of skin hanging on your fingernail. What is that? The cuticle or the cutaneous layer. We're talking about a little flap of skin, aren't we? Cyan, one of the color cartridges in your color printer. Cyan refers to blue color. And cysto is a sac or a bladder-like structure. Cyte or cyto, any word that has cyte or cyto in it is referring to a cell. Dactyl, fingers or toes. If you have polydactyly, you have more than the number of 5 fingers or toes. If you have syndactyly, then you have webbed fingers. Those fingers have webbed together, and we saw an example of that last time when I showed you the fetus going from embryo from 5 to 10. And I told you that it was apoptosis that occurred in between the fingers. And if that apoptosis had not occurred properly, then the child would be born with syndactyly, fingers that are webbed together. D, down from or undoing. Down the hall or upstairs, we have a defibrillator. Right? So if someone goes into cardiac arrest or has issues with the heart, we get the paddles, we go, clear, shock. What are we doing? Defib, stopping fibrillation. Fibrillation is a quivering of the heart. It's not properly working. So we're going to shock the heart and reset the electrical system, hopefully. Dend is tree. When we get to the nervous system and we talk about dendrites and nervous tissue, we will see these tree-like branches or extensions or processes on neurons, and they look like little trees. And demi, like semi, means half. Derm, skin, we're talking about skin today a little bit, and if not today, certainly on Wednesday. Detruse, to force away. your bladder has a muscle around it, kind of surrounding it, called the Detrusor Muscle. That muscle is going to squeeze when you are avoiding, when you're urinating. Dextro to the right of. Most people have their heart on the left side, but there are some people who have dextrocardia. Their heart is actually over on the right side. Their body is basically a mirror image of normal, and everything works fine. It's just kind of a backwards mirror image. It's kind of rare. What is left? If dextro is right, what is the equal term for left? Anybody know? It comes from, if you've had some chemistry, you might have seen this. It starts with S. Sinister. Sinister. So if you're left-handed, you're sinister. Right? Di is 2. Diploid. We've seen diploid already. What does that mean? Diploid means that there's 2 incomplete sets of all the chromosomes. The zygote, the first cell, egg and sperm come together, form a diploid cell. Most of the cells in your body are diploid. They have a normal 2 copies of all the chromosomes. And what process makes diploid cells? Mitosis or meiosis? Mitosis is an actual duplication, right? We know that some errors can occur, but for the most part, mitosis creates 2 equal diploid cells. Dia, through or across. If I'm going to measure the diameter of something, I'm going to measure how far it is across. Or dialysis, filtration of the blood done artificially. Dialysis, you're filtering across a membrane. So that is the term dialysis when the kidneys aren't working. Diastole. We'll see when we get to the cardiovascular system, when we talk about the heart, that the blood pressure, there's a, like 120/80, there's a systolic and a diastolic pressure. The diastolic pressure is the time when the heart is not contracting, when it's not beating. It is a time when the heart is, if you will, standing still. So that's what it stands for, is stand apart or during the relaxation phase. Dips is thirst. If you have polydipsia, you have excessive or great thirst. Dis. In the lab this week, we're going to be playing with a skeleton that is disarticulated. That means that it has everything's been taken apart, right? So dis can mean apart. Dis can also mean opposite. If you look at disinfect, it means the absence or the opposite of infection. Or dissect, right, to pull apart. Dipple, we've already seen di, meaning 2, and dipple even more completely means double. Diuret. Diuretics are medications that cause one to urinate. So diuret means to pass urine. Dorsal, we know, means the back, dorsal fin, or dorsal as a direction. Duct, to lead or to draw. When you think about the ovarian duct or the oviduct or the fallopian tube, they're all the same thing. That tube is sort of leading the egg, or if it's been fertilized, the embryo, down the tube. So it's leading or drawing it down. Or if you think about adduct, we'll talk about joints next week in lab. We'll be talking about muscles and how our body moves. And one of the joint space movements is called to adduct. And that means to move something toward. And then dur. We're going to see that around the spinal cord and around the brain, there's kind of a, I should say, more durable layer, right? Durable, it literally means hard, but it's really more of a durable layer. Think hard, though. That's what its derivation is from. But there's this layer around the heart, kind of a tough outer layer. Dynia is pain. So if you had coccygodynia, you would have pain of your coccyx, tailbone. Dys, painful or difficult or bad. Dysuria would be painful urination. E, out from. Sort of like EX, exit from, or E, out from. And eel, like al, means pertaining to. So we have eel and al, both meaning pertaining to. EF, away from. When we get to the nervous system, we'll talk a lot about efferent things. And that means that it's moving away. Ferent means to carry. We'll see that coming up. And E means away from. Ecto, on the outside. Ectomy, to cut out. Appendectomy, to cut out or remove the appendix. And if you have edema, EDEM, or ED, that's referred to some sort of swelling. If something ends in L, then it means small. So the organelles are what? Basically small organs, small little building parts of cells. EM or EN both mean in or into. Embol means stopper. An embolism is literally a blood clot. It's stopping the blood. And emesis, vomiting. If you have hematoemesis, you're vomiting blood. Right? Dysemesis would be bad or painful vomiting. I don't think there's good vomiting. Emia, condition of the blood. So if you have bacteremia or anemia, some condition of the blood. Encephal, what did cephal mean? Head. Encephal, what's in your head? Your brain, right? So encephal refers to the brain, cephal refers to the head. Encephalitis would be inflammation of the spaces around the brain. Endo within, entero, your intestines. Enteritis would be inflammation of your intestines. And I'll finish up with this one today. Epi, we know this one. Epidermis is on or upon the dermis. Erythro, your erythrocytes are your red blood cells. Estha, sensation. Anesthesia, the absence of sensation. And finally for today, EU, meaning good. You can also put next to that true. So good or true. So eupnea is good, breathing. You see the PNEA. That's breathing or air. If you give a eulogy, you're giving a good word. And you may have heard of eukaryotes, cells with a, quote, true nucleus. So EU can mean good or true. You can add true to that. So that's the first half, and we will finish up the last 10 on Wednesday. It'll bring us through histero, and those 20 cards will be on your exam next Monday. How many of you have had a chance to already make your cards up? A few of you, good. It's something you can do. You can just be flipping through the cards. I hope you found that to be an effective way of studying. If you have a different way, that's fine. But if you struggled with the vocab, I definitely recommend flashcards, and that usually is a way that students find very successful. Now, having gone through the vocab, what questions do we have? Let's think about what we've done. 2 weeks ago, I mean, we've been working on this content actually for a while. There was a makeup lecture for you to listen to about organelles, about cells and organelles. And then we started talking about mitosis. And I mentioned a little bit about cancer and about cell cycle. And that led us into a conversation about transcription and translation. I'll finish that up today. And then, like I said, we're going to be moving into a conversation on histology, the study of tissues. We'll review quickly epithelial and connective tissues. And you'll see for the first time muscle and nervous tissue, just a little bit about it. And then we'll move into skin next time. So in that conversation so far, mitosis, meiosis, cell cycle, transcription that we've had so far, is there anything that needs to be clarified or restated for you? Mastering is going to help you with this. I'm going to brag on the students who just left lab. Good lab scores, good lab quiz scores. Okay, and I hope that's going to continue for the week. Now, much better than typical. No, I'm not going to put all the credit. You guys are working hard, no doubt about it. And I think that's why the quiz grades are good. But I also think this semester, for the first time, I'm requiring more online stuff before you come to labs. And I know that's tedious, and I know there's a lot of time involved with doing those pre-lab activities. I also know so far it seems to be paying off. You're coming in, so far what I've seen, you're coming into labs as a group, more prepared. You're doing better on the quizzes, and that's worth it. Okay, so I think it's worth it. I know it's painful sometimes to do all those extra online assignments, but it seems to be paying off, and as long as it pays off, at the end of the semester you'll be thankful that it paid off with higher grades. So keep that in mind when it's midnight and you're cursing my name under your breath about this is, all right? Keep it in mind. It is helping make you be more successful. So anything at all, histology, or not histology, mitosis, meiosis, transcription, translation. Let's keep going, okay? And then if there's anything that pops up, let me know, please. And it's been a long 5 days. Since I saw you last, I've been down in Tennessee at a horse show, so I've forgotten a few things. Seems to me, oh yeah, let's not forget, there will be a short written section on this exam. It's going to include these words. I will tell you more about it again in a moment. We've already used some of these words in preparing for the lecture last time, and I will be finishing up some of these words today. So I'll tell you more about this on Wednesday, but if you want to, you can certainly start practicing this. These are the same words that you'll have on yours, I'll give them to you. You don't have to memorize the words. And you have to create no more than 3 sentences using at least 10 of these words. That means one of the words that you don't like, don't use. That's fine. And you cannot say something like, in Dr. Mack's class, we learned about transcription, the nucleus, DNA, translation, proteome, cytoplasm, ribosomes, protein, rough ER, genetic code, and mRNA. That will not be an acceptable sentence. Okay? It's been tried and not well. But if you imagine that you are writing, you know how at the end of a chapter, you will see that the author or the textbook company has created some review sentences that very succinctly link words together, and usually those words are in bold as you read those review sentences. That's what you're creating. You're basically creating a review statement, 2 or 3 sentences, using these words. So I don't want 10 sentences. Transcription is the process of. No. I want you to link these words in a meaningful way. So you could say something like what? Transcription, the process of making mRNA from DNA, occurs in the nucleus. You just used 4 of those words, and I'm happy. Okay? As you're using those words, what did you just say? What did you just say? Write it down. As you're using those words, underline it. Okay? So underline the words, because what I will do when I'm grading this is the very first thing I will look for periods. And I'm going to make sure there's no more than 3 periods, right? Because what's a sentence? Something that starts with a capital letter and ends in a period. I'm not going to judge if it's a run-on. I don't care about it if it's a fragment or a run-on. But I'm looking for a capital letter that starts it. I'm looking for a period that ends the sentence. I will look for no more than 3 periods. And then I will look for 10 underlined words. And then I will read it for accuracy. And as long as I see no more than 3 sentences that you've underlined all the words, and when I read it, I don't go, this is an interesting version of biology. As long as it makes sense, then you're going to get all the points. So that's how I want you to approach this. The word that you don't like, the word that doesn't easily link into your sentences, simply don't use. So more about that on Wednesday. Please, if you have any questions on that, let me know. And then we talked a little bit about DNA structure, Watson and Crick. We talked about mutations, right? That during replication, the DNA is replicated, it's copied, and it's done rather quickly. And in that rapid copying, there are some mistakes made. And if those mistakes are not corrected, then the cell is stuck with them, and we call that a mutation. And those mutations can accumulate over our lifetime, leading to increased risks of cancer. And then we talked about transcription a little bit, and we had this picture here, right? This overview. Let me start here with you. What slide is this? What page is this? Anybody?
56.
56, page 56. Just to get us started, we'll ramp into this. So what we see here are really both processes laid out for us. On the top, right, DNA, which is found where in the cell? In the nucleus. In your cells and my cells, that DNA is really how many molecules? 46, right? Long molecules of ACTs and Gs, right? All linked together, wrapped around proteins. And those things are called chromosomes. And this is transcription. When in the cell cycle does transcription happen? I never asked you this before, but when does transcription happen? G1? G2? What do you think? G1 and G2. Because what is G1 and G2? Those are the gap or growth phases. That's the time during the cell cycle where the cell is doing what cells do. And this is something that cells do all the time. So they're transcribing their genes. They're making proteins. This is happening in G1 and G2. Can we see the chromosomes under the microscope during G1 and G2? No, that's that interphase time. So when you imagine this, what's the state of the chromosomes? They're kind of unwound. They're not tightly wrapped like we see during mitosis. They're unwound. And during that unwound state, this is what happens. This is extremely simplified, but it's where I need you to be for this course. So in the nucleus, we're seeing a little tiny glimpse of one little part of a chromosome. And we see that double-stranded DNA, right? It's red. Just a little piece. remember that the DNA, two strands, are only held together by some hydrogen bonds. Remember, the C's and the G's bind with 3 hydrogen bonds. The A's and the T's are only held together by 2 hydrogen bonds. So these 2 strands can be rather easily unzipped. That's what we're seeing here. The 2 strands of DNA have been unzipped, and there's a lot of machinery, a lot of enzymes that are involved here that are not part of what you need to know, but these enzymes take the double-stranded DNA and make a single-stranded RNA. That process is called transcription, right? We are transcribing. In fact, going back to the sentences, I think I have the word transcription. If you want to instead change it to the verb transcribing, I'm okay with that, right? If you want to change the word translation to translating, that's okay. You can change the tense of those words if it fits better in your sentences. Now, this process of transcription happens where? In the nucleus. It has to be there because that's where DNA is found. Then what does that single-stranded mRNA do? Leaves the nucleus through the nuclear pores that you can see in this cartoon, and that single-stranded molecule kind of squiggles through the nuclear pore and then bumps into, very purposefully, bumps into what? Ribosomes. Now those ribosomes sitting here, this purple dude, the ribosomes here, we just see one ribosome. There'll be thousands of ribosomes. So here we just see one. Those ribosomes could be freely floating around in the cytoplasm or could also be bound to a series of membranes called the rough endoplasmic reticulum. Again, we don't see any RER here. We just see a lonely ribosome waiting in the cytoplasm. And this mRNA is going to bump up to this ribosome, and the ribosome is going to say, my job is to do what? My job is to translate. I'm going to take the message, the signal, the mRNA, the messenger RNA, and I'm now going to translate that into a protein. The protein, in order to make the protein, there better be in the cytoplasm plenty of the free amino acids. And so these little green hexagons or little squares represent those 20 different types of amino acids that need to be there from your diet, or your body has made them. And after the ribosome has read down, it kind of scans along the mRNA, it makes the protein. And then what does that protein have to do? As the protein is made, it's a long chain. We would call that the primary transcript. It's just a long chain of amino acids. And we know that those amino acids are going to interact with one another and eventually fold and twist and come to a final 3-dimensional structure. So that's what's shown here. We see at the bottom of the picture sort of the long chain of amino acids has folded in some unique way. And every protein has its own unique shape, which contributes to its unique function. Okay, so that's the big idea. And that same idea is shown here in this second picture, where 1 is transcription and 3 is translation. Now I need a little bit of help. Where did I end last time? I think, now I remember. What are these little orange lunar landing molecules called? Those are called tRNAs, transfer RNAs. And what are they transferring? Carrying. They're carrying the correct amino acid to the ribosome. Now, how does this whole thing work? The tRNA is made of what? It's an RNA molecule, right? Don't make this harder than it is. It's a molecule made up of A, C, Gs, and Us, right? It's an RNA molecule. And that RNA molecule is going to bind to and recognize a portion of the mRNA. So look right here at this little boxed area. So what we have in here is that there are three little ACGs and Us. 1, 2, and 3. Those represent what's called the anticodon. So those 3 little letters, ACG or U, whatever they are, represent the anticodon, and they bind specifically to the 3 letters on the codon. So I know it's messy up there, but I've got yellow sitting over the 3 letters of the codon, blue sitting over the 3 letters of the anticodon. They come together. That's where the specificity comes in. And notice that depending upon whatever those 3 letters are on the mRNA, it brings in the correct amino acid. In this case, a green one, which would be one of the 20 amino acids, just shown in cartoon. And this is where we ended up, as I recall. So let me introduce this, and then I'll go back for questions. Across the top, in red, it's one of 2 strands of DNA. We don't see the other strand, but it would be associated there. And the DNA was read, and through the process of transcription, what happened? We made mRNA. G matches up with C. C with G, and A with U. Remember, this is an RNA molecule. So the DNA across the top, you see T's in there, don't you? But we're making mRNA. mRNA is made up of A, C, G's, and U's. So you don't see any T's in the mRNA in the yellow sequence. You see U's instead. And A tells the mRNA, in the process of transcription, tells it to make a U. Continue that process. And then that mRNA is going to go out into the cytoplasm and hit a ribosome. That ribosome is going to then translate the mRNA and make protein. And every time the ribosome sees CGU, it is going to know to put in this amino acid called arginine. And every time it sees next UCA, it will know to always put in the amino acid serine. And on it goes. Now, you're not going to memorize that. I will give you a table that gives you, quote, the genetic code that helps you understand, like a highlights for kid kind of activity, a decoder ring, how to figure this out. I saw a hand in the back, and then we'll go forward.
So what happened to the mRNA after translation?
It's still there and can be used again and again and again as long as it hangs around. A little outside the scope of the course, but mRNAs, some of the times they hang around for a long time, sometimes they hang around for a very, very short time. As long as they're there, the ribosome can grab it and make another protein. Okay? So I mentioned this already. This is that long chain of each of these little boxes represents another amino acid. You're not responsible for knowing the names of the individual 20 amino acids, but those are their 3-letter abbreviations. And you see we make a long chain, and that is called the primary structure, right? That's the primary structure of the protein. That's what the ribosome just made. And then, as I've already said, each of those amino acids is going to interact with each other in a unique way, causing the protein to twist and turn and create a unique structure. Now, there are 3 different kinds of RNA in this story. There's actually dozens of different kinds of RNA in your cell, and we're learning more and more about them all the time. But for the purposes of this course, 3 mRNAs. So let's get them straight, and this table will help you. Number 1, the one we talked about the most, mRNA. It carries the message. It is what is made from the DNA in the process of transcription and what will be decoded by the ribosome in translation. In all of our pictures, by chance, it's yellow. Then I've got a crisscross here. I apologize. The bottom one, we have already talked about transfer RNA. Transfer RNA is the one that carried the correct amino acid into the ribosome, and it looked like a little lunar landing molecule, didn't it? Now, these 3 little letters right there sticking off, what are those called? In that box, those 3 little things sticking out are part of the RNA structure. They are the anticodon. So the tRNA carries a 3-letter sequence called the anticodon. Where do we find tRNAs? In the cytoplasm. Why? Because that's where they're useful. That's where translation occurs. Then lastly, and I have not discussed these guys before, ribosomal RNA. Sounds like an RNA that must be really important for ribosomes. Ribosomal RNA is made in part by the nucleolus. Well, let's think back for a second. Where's the nucleolus? It's that dark staining center in the middle of the nucleus, and it's there that the ribosomal RNA is made. So guess what? That's where your ribosomes, part of your ribosomes are made. This is another reason why you need those nuclear pores. You need mRNA to get out, but you also need the building blocks for ribosomes to be able to get out of the nucleus to the cytoplasm, because it's in the cytoplasm that the ribosomes are useful. So ribosomes, what are they? They are made up of ribosomal RNA and proteins. They're little combinations of the two. You need proteins. You need ribosomal RNA to make proteins, and you need proteins to make RNA. So this is the chicken and the egg question of molecular biology. Questions on either of these processes? What's been described so far? This is a 30-foot view. It's just a big overview. This is not detailed, really. It's just a big overview.
So where does the what made of the tRNA before it actually binds to the ribosome to bring the correct?
I missed the first part.
What made up?
Oh, okay. So there are genes for tRNA, right? So in your DNA, there are sequences. Rather than making mRNA, you make tRNA. Another time. So somewhere else along your chromosomes, there are genes for tRNAs. Okay? And those tRNAs are going to make, those tRNA genes are going to make tRNAs. And those are going to be sitting around floating in your cytoplasm. Okay, so I said this last time. Here it is in words. This may help some students. Some students just get confused between transcription and translation. Again, you've got to go out about 5 or 6 letters before they become alphabetical, but they are alphabetical. Transcription is going from A, C, T's, and G's to A, C, G's, and U's. It's the same language, if you will, right? We're just changing one letter in the alphabet. It's the same language, the same alphabet. But when you translate, you need to completely change the alphabet. It's like going from, English to Mandarin or something. It's a completely different system. And so rather than having a alphabet with only 4 letters, A, C, T, and G, now you go to an alphabet with 20 letters, right? 20 different amino acids. And this is part of the magic of Watson and Crick. When they put their little tinker toy model of DNA together, they said, aha, now we know, or at least we hypothesize how this works. And for the next 20 years, scientists proved what Watson and Crick hypothesized. That is, that DNA going to protein was based upon an mRNA intermediate and a 3-letter code. Okay? And it was a really long story. We talk about it now like, everyone knows this, but it was, it's only details that we've understood the last 60 years. So it's not like it's old news. Now here, again, what Watson and Crick told us is that the genetic code, how DNA makes protein, is a triplet. It's a 3-letter code. And what they hypothesized, incorrectly, was that 2 was not enough and 4 was too much. And so they said, you know what, this whole thing must be based upon 3 letters. So they were right. Don't worry about the math here, but 3 is the magic number. And this is the code, this is the table that I will give you. This table can be called the genetic code by some books. But you'll also hear the genetic code referred to as basically all the DNA that's in you, right? Your cells contain the genetic code. So you'll hear the term genetic code used in different ways. But this table can be considered the translation genetic code. What you see here is that every 3 letters of A, C, G's, and U's always will tell the ribosome to put a particular amino acid on the growing protein. Every single time, for example, the cell sees CCC, the ribosome is going to instruct it to put a proline on there. Every time it sees CAU, it's always going to tell it to put a histine on there. Every time it sees AUU, it's going to be an isoleucine. Don't worry about the names. You'll have the table. Of these, there are 64 possibilities. Now, how many amino acids are there? 20. So, there's a little bit of a wiggle room here when we go from 20 amino acids to 64 different codes. So what you will see is that there is some redundancy. In other words, there's 4 different ways for proline, right? You can have CCU or CCC or CCA or CCG. So there's some overlap, if you will, but there's no doubt every time the cell sees CCU, it'll always put in proline. There are 3 that act as stop codons. In other words, they're like the sentence. They tell the ribosome that you're done. When the ribosome comes along and sees UAA or UAG, then it's like a period at the end of a sentence. The ribosome knows I'm done. It falls off the messenger RNA, and the protein is done being made. So we've got the period, and there's also a capital letter. Like I told you, when I'm reading your sentences next week, I'll be looking for capital letters. AUG is like screaming at the ribosome saying this is where to start. So like when you're reading, you're looking for those capital letters to start your reading at the capital letter at the beginning of the sentence. So too the ribosome is scanning along and looking for that capital letter. And the AUG is considered the start codon. So there's only one start codon, and then there are 3 of these triplets that count as a stop, or like a period. And I'll give you this table, and we're going to practice right now how to use it. This is the same picture I showed you earlier. And I told you that every time the ribosome sees CGU, it puts in arginine. Well, let's go back and look. CGU, go back. Find it on here. CGU, where is it? CGU, there it is, right? So every time the ribosome sees CGU, it's going to put in ARG, right? No doubt. I'll give this table to you. You just got to figure out how to read it. Let's look at the second one. Every time the ribosome sees UCA, it's going to add in this amino acid, SEER. Let's go back. UCA, there it is, UCA, SEER. So that's where that's coming from. It's not random by any means. It's very well programmed. And I'll let you do the other 2. But every time it sees UGG, it's going to be a trip. And every time it sees ACU, it's going to be a 3. Always, always, always. Now, there's 3 words that I want to share with you about the genetic code. It is said that the genetic code is universal. That means that almost, and there's very few exceptions, but almost every living organism on the planet, bacteria, animals, parasites, you name it, use the exact same set of rules. There's a couple of weird little worms in Africa that have a different set. But for the most part, everything else on the planet uses the same universal code. That's why it's called universal. There's 2 other words about this genetic code. One, it's redundant, but it is not ambiguous. I've already introduced this. Redundant, if someone is redundant, what does it mean? Man, you're redundant. Keep saying the same thing over and over and over. In other words, there's more than one way for me to say proline. Remember I told you there's 4 different ways for me to say proline. That's redundant, right? I could sit here and say CCU or CCC. They all mean proline. That would be redundant for me to say all 4 of them. But the code is not ambiguous, meaning that every time the ribosome sees CCU, there's no doubt, right, that it's going to put in a proline. So even though there's some redundancy built in, there's no ambiguity. It is an absolute perfect system. Now, what does this suggest to us if there's a mutation? Look at this. If I changed these letters up here in the DNA, that means that my mRNAs could be different. And if my mRNAs are different, then my protein might be different. And if my proteins are different, if my amino acids are different, that means that the protein may not fold or bend or act in the proper way. Okay, and that's the whole basis. That's kind of our take-home lesson as we get to the end of this about mutations and why we care about mutations. Now, here is another version of the same thing. I promise you I've not changed anything around, just a different version of it. So tell me right now, what amino acid, if you were the ribosome and you saw UUG, what amino acid would you add into the growing protein? UUG. So find it. UUG, I see it up in the upper left, right? UUG, Leu. Okay, it's leucine. Don't worry about leucine, just Leu. And how about CGG? CGG, ARG, right? So we get the idea. That's what you need to be able to do. You need to be able to read the next three letters and from it figure out what amino acid's coming in. What structure carries in the correct amino acid? The tRNA, right? The tRNA is what carries it in. So because the genetic code is nearly universal, this is why we can do genetic engineering. This is why we can take, for example, the firefly gene that makes fireflies glow, and we can stick that same gene and put it into a plant. And now the plant will glow. Okay, so this is a glowing plant on the very top. We can take that same gene and we can put it into a cat. And now the cat will glow. Now, on the bottom, you've got a glowing kitty. Okay, so under fluorescent light, this little kitty will glow. And here you can see that's all an internal glowing because, the cat's got this firefly gene on. This is why we can take a heat-resistant gene from some rice organism and stick it into corn and now make that corn plant heat-resistant. Or we can take a pest-resistant strain of corn and put that pest-resistant gene into tomatoes and now make those tomatoes pest-resistant. These organisms are under a lot of controversy, right? These are now genetically modified organisms, GMOs, and this is creating all sorts of hoopla. Most of the food you eat probably has been genetically modified, okay? And that's just the way it is, right? In order for us to feed the billions of people on this planet, we've got to figure out a way to increase our crops to resist, disease and pests and drought, if you will. So genetically modifying organisms is certainly going to be a part of our future. Is it dangerous? Is it dangerous for you to consume a GMO, genetically modified organism? Absolutely no evidence thereof. But there are people worried about it. Now, do you consume DNA of plants? Yeah, right. When we eat lettuce, we're eating the DNA of the cells. We're eating DNA. When we eat a modified lettuce plant, are we eating modified DNA? Sure. Okay. But is there any evidence that it's harming us? There really isn't any evidence of it. But there's a big market right now, isn't there? Non-GMO. We'll see, right? My take on it right now is that there's really no tremendous evidence to be concerned about it. And if we start insisting on non-GMOs, then half the world's going to starve because we won't have the food to feed them. But that's the other side of the argument. Question? None. No difference. I mean, a protein's a protein. And these genes, most of the cows that you're consuming, right, beef, has either been injected with hormones. Well, that's not good. So now we're going to genetically modify them to make more meat per, muscle or whatever. So we're going to make them grow up faster and have more muscle on them. So it's a trade-off. But there's no evidence that the meat from the animal is any different than meat from a non-genetically modified organism. Okay. So when we say GMOs, we're not talking about just plants. It could be animals too. So the chicken you got from Tyson, if you go to the Dominican Republic and you look at a chicken, it's the most anemic-looking thing, little tiny bit of meat on those bones, right? And then you get the chicken from Tyson. It's a meatier bird. That bird's either been given more nutrients, stuck with some antibiotics, or is genetically modified to grow bigger, faster, and stronger, so that we can, kill it and have more chicken faster. There's no evidence that there's any problem with that so far. And these glowing kitties are really cool. They even found out that the hair gets in the way of the glowingness, if that's the right word. So now they put this same gene into those nude kitties. Have you seen the nude cats? The hairless cats? They look like a Shar-Pei, but they're a cat. And they glow really well. Right? They don't have any hair in the way, so they glow. They're really cute. They purr, and they like a little night light. And There's now glowing kitties and glowing dogs and glowing pigs and glowing whatever. You can look on the internet and find them. And could we have glowing humans? Easy. Should we do it? Don't think so. But could we do it? When you go to Meijer and you see those glowfish, have you seen those little glowfish? Really cheap, like the bright neon pink and orange and purple and green neon fish. Those things aren't normal. Those things aren't natural. Those have been genetically modified. So they've stuck a gene like this into their little embryos, and when that fish grows, it glows a different color. But those are not natural fish. But they're pretty to look at. Okay. Now there's one more little piece to this that I want you to appreciate, and that is reading frame. I told you that I will be reading your sentences looking for a capital, for a capital letter and going all the way until I see a period. The ribosome needs to do the same thing. The ribosome needs to know where to start. I told you there was a start codon, AUG. And the ribosome needs to know where to stop, where the period is. And I told you there are 3 different versions of a stop codon. And the ribosome is limited to reading groups of 3, right? This codon idea, this triplet idea. So the ribosome starts and only looks at 3, then the next 3, then the next 3, then the next 3, then the next 3. That's all a ribosome does. It doesn't get creative and do groups of 1, 2, or 4 or anything else, just groups of 3. So there is this idea of reading frame, and let me try to tell you about this. So if I had this sentence, the big red dog, right? So that's a sentence, right? And it starts off with a capital letter, so I know it's the start. And then I see groups of 3. And then I see a period. What if the ribosome did not know where to start? What if instead the ribosome started not on the T, but started on the H? What would the ribosome see? Right? Junk. So it's very important that the ribosome knows where to start and that it stays in what we call the reading frame. 3, 3, 3, 3, 3. So I'll come back to this idea in a second. So that's the reading frame idea. The ribosome needs to know where to start. And if there's an error in the reading frame, then that can also change the entire protein. Like I just showed you, if it started 1 off or 2 off, it would be way off. Now, what if instead the ribosome started, what if instead it jumped over, let me do that again, sorry, the big red dog. What if it jumped over 3 letters? Let's say it jumped over all 3 of those letters. Now the sentence, the protein would be what? The big dog. Did that change the meaning? Yeah. Did it change the meaning significantly? Maybe. Maybe that it was red was really important, or maybe the red was an insignificant feature. So we'll see that depending upon which amino acids are changed, it may or may not make a big change to the protein. It might be devastating, it might be minor, but there's definitely been a change, no doubt about it. So let me go back and just remind you about these tRNAs. What are they again? There's these little lunar landing molecules. And what do we call these 3 letters right here? Those 3 letters are referred to as the anticodon. And what do those anticodon letters, A, C, Gs, and Us, bind to? They come down and they look for the next 3 letters on the mRNA, and those 3 letters are called the codon. So the codon and the anticodon talk to each other. So what we see here is here's the big ribosome, right? The big ribosome, big purple dude. And the ribosome's kind of going along the mRNA, going in one direction, and now there's 3 next letters. So whatever those 3 letters are going to bind to a correct tRNA, and that tRNA is then going to know to carry in the correct amino acid. So we see that here. There's those words again for you. Oops. Here are those words again for you. Anticodon and codon right there. Right, anticodon and codon. Now, a ribosome has a big and a small unit. Nothing fancy about it. It's like 2 little pieces that fit together. Again, what are these ribosomes made up of? partly ribosomal RNA and partly proteins. And those ribosomal RNA molecules are made in the nucleolus. Remember, we talked about that. So we need to have these two parts, the big and the small unit of the ribosome, to work. And here, we've taken off the top part. So we just got one part on here. And here's the very first AUG. What did I tell you? AUG was what? The start. So the ribosome's kind of reading along, looking for the first AUG. When it finds the AUG, it says, okay, that's where I'm going to start translating. I'm going to start reading at that place. And the AUG, look above it, will match up with this tRNA that has UAC on it. What does that UAC match up with? U matches up with A with U, and C with G. That's where the magic is. That's where the translation, the decoder ring is, right there. And every time that matches up, look what amino acid's attached to that tRNA. At the very top it says MET. That will always be the first amino acid in a protein. I'm oversimplifying this a little bit, but we'll start there. Okay? That's all we need to understand. Then the next three letters, whatever those are, here comes the next amino acid. They tell the protein to build in leucine. And on we go. Don't worry about the E, P, and A. I won't worry about that this semester. So, mutations. What are mutations again? Changes in the DNA. How can those DNA changes occur? Either from mistakes made in replication, right? There's always mistakes made in replication, or perhaps some environmental issue, X-rays, some sort of carcinogen, something comes along that mutates our DNA, or there could have been an issue, like a big-time issue, like with chromosomes. Like if I have an extra whole chromosome, that could be like Down syndrome. That's another kind of mutation that I'm not dealing with here. What I want to deal with here are just point mutations. Single little changes in the ACTs and Gs of DNA. Now those mutations can come in three different main types. There can be insertions, simply meaning that there's an extra letter added in to the DNA. There could be deletions, where letters, ACTs and Gs, are lost. Or there could be substitutions, where a C is replaced by a G, or an A is replaced by a T. Now I am talking about DNA there, right? So I use T's. There's a mutation in the DNA. So let's take a look at what happens. At the top is a normal mRNA sequence. What are we not showing here? We're not showing the DNA. So the DNA is not in this picture. We've already made the mRNA. I know it's mRNA because it tells me it is, but I also see a lot of U's. So I know it's RNA. The top represents the mRNA. We can go back to our genetic code, that table, and see that every time the ribosome sees AUG, it'll put in MET, methionine. Every time the next three letters the ribosome sees, AAG will always be lysine. The next three, UUU, will always be phenylalanine. At some point, the ribosome gets to a sequence called UAA. That represents one of the three stop codons. That's like the period. There is no amino acid added. It's simply like the ribosome pops off and the message has been made. Now that's a very short protein. It's only made up of 4 amino acids, but you get the idea. Now what if instead there has been a mutation to this gene? That mutation in the gene has caused a missing letter in the mRNA. And what's been lost is we've lost a U. Okay, so the mRNA does not have the U. The ribosome still sees the AUG and starts translating. It puts in MET. The next three letters haven't changed. We still see lysine. The next three letters, though, rather than being UUU, are now UU. The U's missing, UUG. UUG, go back to the table, puts in a different amino acid, leucine. The next three, CGA, that look at the table, gives you alanine. And it keeps going. What's happened? We've lost our reading frame. We're no longer reading in the proper groups of 3, and we're no longer going to see the stop codon, are we? So the ribosome is going to keep reading, and most of what it keeps reading is going to be complete junk. It might be like some of your sentences, right? Just kind of keeps on going, makes no sense. No, hopefully not. So that would be a devastating protein, right? That protein would have little to no use for the cell. What if instead there was an extra down here, there's an extra U stuck in as an example? What would happen to this protein? If there's an extra U stuck in, look how devastating this would be. You start okay, the ribosome sees AUG, puts in methionine, and then because of that extra U, the ribosome now sees UAA, which is one of the 3 stop codons. So as soon as that protein started, it got stopped. If that protein was critical to the synthesis of energy, or some very important protein for the cell, that cell can no longer survive, can it? So what's that cell going to do? Apoptosis, right? I told you that when mutations occur, there's one of 3 solutions. One, fix it. That would have had to have happened when it was put in and someone came along and fixed it. Or die, right? That would be what happens here. If this was a really important gene product, a really important protein, and that cell could no longer function, then the cell would essentially die, undergo apoptosis. And thirdly, if it could live with the mutation, it would just survive, wouldn't it? It would limp along in a different way. Here's another one. Now, what if instead of a single letter, we pull 3 out? And I've already mentioned this already a little early. What if I pull out the A, and the G? I just happen to pull out 3. That means that as the ribosome reads down along, it's going to miss a whole amino acid, but it's going to be able to keep on going, and it's still going to stop in the right place. That would be like going back to my sentence and taking out the word red. Would that protein that it makes still be useful? Maybe, maybe not. Maybe that word red is critical to the functioning of the protein, or maybe that word red is not so critical. So this protein that is made may or may not be good. It may be mutated. It could cause disease in a person if that's the product that they have left. And then here's another one. Here is a substitution. Now, rather than adding 1, taking away, or taking away 3, now we're simply going to substitute. We're going to substitute this C and put in instead a U. Now, look what happens here. The ribosome still gets started at AUG. It still has lysine. It still has phenylalanine. GGC at the top now becomes GGU below. If you go back and look at the table, this is where that redundancy comes in. You will see that CCG and CCU, or GGU, both stand for, or both code for, the same amino acid. Did a mutation occur? Was there a change in the DNA? Yes, right? There was a C change to a U. Is that a change? Yes. Is that a mutation? Yes. There's a mutation in the DNA. Did it have an effect? No. Okay, so this would be called, this word's not on here, this would be called a silent mutation. Okay, silent mutation. Because was there a mutation? Yes. Was there a change in the DNA? Yes. Did it have an effect? No. It was a silent change, a silent mutation. Now, what feature of the genetic code is being featured here? The silent mutation is because there is redundancy, right? There's more than one way to say glycine. And so the redundancy of the genetic code made no difference in the protein. So it's a silent mutation. Now, here's another one, right? A instead of G. Did this substitution make a difference? Yes. In this situation, it absolutely did. So again, one letter can make quite a difference. Here's glycine becomes serine. May it be a critical difference? I don't know. It could be majorly different. It could be, it may not have any effect at all. But a mutation definitely happened. There was definitely a change in the amino acids. And how devastating it is, we don't know. Now, why do we care about this? Here is a disease that perhaps you've heard of, sickle cell anemia. On the top left is the wild type. Wild type's a way in genetics of saying the normal, the most frequent. So the most frequent version of Hemoglobin. Hemoglobin is the protein that carries oxygen in your blood and is in your red blood cells. And the normal sequence has a CTT in it. That's the DNA sequence. That CTT is transcribed into GAA. And that GAA is then translated to a amino acid called GLU. In the most common version of sickle cell anemia, the T over here on the left is changed to an A. It seems small enough, one little tiny letter out of the billions in your genome. That CAT then transcribes to GUA, and that GUA now translates to VAL. It's a devastating change. Life still goes on, but people with this mutation have sickle cell anemia. Here's what happens. They're red blood cells, and we know what red blood cells look like. On the top, these are normal-looking red blood cells. They look really happy. On the bottom are red blood cells that are in a sickling episode with a person with sickle cell anemia. And what you see is that the cells are very elongated. They're sickled. Here's what happens. This change in the ACTs and Gs, the mutation, causes two distinct things to happen. Number one, these hemoglobin molecules don't carry oxygen as well as they should. Therefore, the person is anemic. That's where the anemic part comes from. So the oxygen is not carried as efficiently as it should be. It doesn't bind as well. Number two, sickling. During times of stress or during times of high altitude, these folks cannot go on planes. These folks could not be climbing Mount Kilimanjaro. The hemoglobin molecule, rather than just being all over the molecule, starts to polymerize. In other words, they make a long chain of each other. Right? So this molecule starts sticking to each other, making a long chain. Now, you already know that red blood cells are squishy. They can change shape. You've seen them crenate, and you've seen them get larger. So you know they can change their shape. These hemoglobin molecules then cause a reshaping of the cell. Those little red blood cells can no longer go through your capillaries. They're too big, and they cause blockages in the blood flow. So these individuals have very, very painful episodes where blood flow is reduced or stopped to organs. Now, if it's stopping blood flow to your finger, you're going to survive. It might be very painful, but you'll survive. If, however, you're stopping blood to your heart, your liver, your kidney, or some other vital organ, this could be life-threatening or life-ending. Okay? And that's what happens. These individuals have, the more sickling events they have, the more chance the blockages will occur in major organs. And over time, they experience tissue damage, damage to the kidneys, damage to the liver, damage to the brain, and they live with those consequences. If a person with sickle cell anemia goes into the hospital, they're going to give them blood thinners, and they're going to give them transfusions. Let's get as many of those nasty, wrong red blood cells out of there, and let's try to thin the blood so that blood can get through those clotted areas. So, one little letter, right, in the DNA makes a disease state. So I wanted you just to kind of get to that end point and appreciate that. And there are dozens of diseases that are all about one letter. All right, one letter changes. Cystic fibrosis is another one. There are a lot of diseases, very devastating diseases, that are a result of one single letter change in your DNA. So, and that's just another example that we've already seen before. So any questions on this whole transcription translation? Why do we care? That's what I really want. I want you to come back and realize we learned this because there's, really cool stuff to learn about. Diseases are cool as long as it's not you. Right? They're fun to talk about, fun to study. It's not fun when it's you or your family member, but they're interesting. So, questions on transcription or translation. That brings us to the end of that whole chapter. It's a long chapter, right? Cells and organelles and transcription and translation, and I threw in some cancer and some cell cycle stuff into it as well. Let's review those organelles, shall we? Don't look down, because the answers are in front of you. Look up at the screen. And what is number 1 pointing to? Nucleolus. Not only do I want you to tell me the organelle, but tell me its function. So the nucleolus is important in the making of ribosomal RNA, right? The making of ribosomes and the components for ribosomes. Number 2, the nucleus. What's sitting in the nucleus? The chromosomes, right? The DNA. Number 3, that is pointing to the little tiny dot. That would be one of the ribosomes sitting on the rough ER. And the ribosomes are all about making proteins, right? Protein synthesis, translation. Number 4, who knows? It could be a lysosome, a peroxisome, some other vesicle. You can't tell them apart by looking at a picture like this, so it's some unknown vesicle. Number 5 is pointing to the membranes now. That would be the entire rough ER, again making proteins. Number 6, another series of membranes a little further out. Golgi, that's all about what? Packaging, right? It's receiving all the proteins made by the rough ER and packages them and makes lysosomes and makes those little vesicles. Number 7, that little string, one of the cytoskeletal proteins, an actin, some sort of molecule that helps give the cell shape. Number 8, a series of membranes connected to the rough ER but have no ribosomes on them. That is the smooth ER. The smooth ER is responsible for making. It doesn't make proteins. There are no ribosomes there. It instead is responsible for synthesizing lipids. Steroids are made on the SER. Number 9, mitochondria, all about the making of ATP. Number 13, the two of them together make up a centrosome. Each of them individually is a centriole, little ole. And those little dudes are going to, during prophase, are going to do what? Separate. One goes to the North Pole, one goes to the South Pole. And from them are going to spit out mitotic spindles, grab the chromosomes, and pull the chromosomes apart during anaphase. Number 10, who knows, right? Another one of the vesicles. Number 12, again, is calling a lysosome. I don't know. Some other vesicle. I can't tell it. And then 13 would be what? Inside the cell membrane would be the cytoplasm, right? Or it could be pointing, I don't know, to the plasma membrane. It's kind of hard to tell what it's pointing to. The plasma membrane separating the inside from the outside, and everything inside being called the cytoplasm. So that's a quick review on the organelles. It's a big part of this test. I want to move on to chapter 4 right now, and that is on histology. A lot of it's review. You've already seen 6 connectives and 6 epithelial tissues in lecture or in lab, and I'm going to add to that understanding a little bit. So hopefully a lot of this is just to sit back and relax a little bit and confirm what you know and maybe make some connections or some things you don't know. I'll pause here just for another second. Any thoughts, questions right now? If you do have any questions, stop me or come on Wednesday with any questions about transcription or translation. Chapter 4, tissues, the study of histology. Now, let's just kind of back up here and let's figure out why we care again. This is so cool. I know when you're a student and you're learning this stuff, sometimes the awe of this is lost because you're having to learn details. But egg and sperm come together, make the first cell called the zygote, and that cell becomes 2, 4, 8, 16 cells. Eventually, we have what's called a blastula or a blastocyst, and that is a what? Blast. Everything in your body will have germinated or budded from that ball of cells, the blastula, blast. And there's one more stage called the gastrula. And the gastrula, if you look at the blastula, what you see is that the ball of cells has become hollow, right? And that is the stage at which you were when you grabbed onto your mother's uterus. So after 5 or 6 days of floating down your mother's fallopian tube, unless you're one of those newfangled test tube babies, this is how it all happened, right? So fertilization up in the fallopian tube, float down for 5 or 6 days. By that time, you were a blastula, and you grabbed onto your mother's uterus. Then, that hollow ball of cells, imagine taking a balloon, and now a good-sized balloon, and now sticking your fist up into that balloon. And as you stick your fist up into that balloon, the cells that were on the outside are being drawn up inside where your fist is going. And you begin to create not just a single hollow cavity, but you start creating 3 layers of cells when you do this. And that's what the gastrula is. The gastrula is the beginning of 3 different layers. And these layers are called germ layers. And the layers' names are as follows. On the outside is the ectoderm. We just saw in our vocabulary that ecto means outside. Derm, layer. In the middle, mesoderm. We saw the mesothelium, right? Epithelial layers that kind of are found in the middle of the body. Mesoderm, the middle layer, shown in sort of a salmon orange color. And then the inside layer, yellow, is the endoderm. We know endo means within. So we have 3 layers, outside, middle, and inside. And in addition to those 3 layers, there's a few other cells called germ cells. These are going to be reserved to someday become your egg or sperm producing cells in your body. Now, depending upon what layer of cells or which layer you're in, dictates what you will become. So let me kind of go through that really quick. The ectoderm, the outer layer of this gastrula, the outer layer, the blue layer, will become the epidermis of the skin. Well, that makes good sense, right? The outer layer of the blastula becomes the outer layer of you, but also becomes your central nervous system. So your brain and your spinal cord are formed from the outer layer. Okay, that's all I want to say right now. So the brain and the spinal cord, what's called the central nervous system, and your epidermis. Have those 2 in your head. Let's go to the inside layer. This makes sense. The inside layer is going to make up your digestive tube, your intestines, and your respiratory tube, down your trachea and down into your airways. So endo, within. So the very middle structures of your body become the endoderm. So think respiratory tube. That is your trachea, going down to your lungs. Your throat, your pharynx, and your digestive system are largely from the inside layer, the endoderm. That leaves pretty much everything else to have been made from your mesoderm in the middle. The mesoderm layers will become your bone and your red blood cells, pretty much all of your connective tissues. So I always just think all of the connective tissues, you know what those are. So fat, blood, bone, cartilage, all that's going to come from the mesoderm and your muscle. So all of your skeletal muscle, all that's going to come. So your muscle and your connective tissues. That's a little bit simplified. There's a few exceptions along the way, but that's a good place to get started. Okay? That's all I need you to know as far as these layers go. Now, as tissues, as we go from a zygote to a blastula to a gastrula, you could say that the cells are becoming more differentiated. Remember the term differentiation? Things are becoming different from each other. So once you're a cell stuck in the mesoderm, your choices are muscle or connective tissue. If your cells are in the outside ectoderm, your choices would be either epidermis or nervous system. Okay? So that's how this whole thing works. Now, let's think about, knowing that much, about stem cells. This topic has gotten a little bit off the front page. It wasn't that long ago that it was a very heated debate. Still is. People were being asked to vote in California, for example, on whether or not they could use state funds for stem cell research. President Bush, the younger, had a moratorium on stem cell research. It's been lifted now. And let's talk about what stem cells are and where the controversy is just for a moment. Make sure we understand this. So when you hear someone like Michael J. Fox talk about Parkinson's disease, or Nancy Reagan years ago talking about Alzheimer's, or Christopher Reeve when he was alive advocating for stem cell research for spinal cord injuries, this is what they hoped. This is what was claimed, that we could take, science could take a a blastula, right? This hollow ball of cells. There's a set of cells in here that will become the embryo itself. So this ball of cells will become everything in your body. Everything. And we could take those cells and we could put those cells into a petri dish and trick them chemically, hormonally, to become blood cells or muscle cells or heart cells or any of the different cell types in your body. Can we do that? Some success, yes. Notice that all of those things say cells. None of those things say heart, liver, brain, kidney. All of those things are cells because you are taking cells from this blastula. Now, where's the controversy? This, right? That's a living human blastula. Now, there's no doubt in my mind, as a scientist, when I look at the life cycle of a frog, I see a frog life cycle starts when egg and sperm are combined, and then we get a little tadpole, then we get a frog, right? And in a human life cycle, same thing. Life begins when egg and sperm come together, and we form a blastula, or a zygote, and then go through the cycles. So there's no question in my mind that life begins at conception. The question is, as a society, is where do we value life? Right? That's really the question we have to be asking as individuals and as a society, not when does life begin. There's no scientific doubt about where that is, folks. Right? That's just hogwash. But the conversation is, where do we value life? And at what point do we begin to value life? And do we put protection on life? The problem is here, we're only creating tissue or cells. Why do you think we can't, now this is now, do you think we'll ever be able to make a heart? Why do you think at this point we can't make a heart or a liver or a kidney by doing this? There has been some limited success in making skin. Okay, we can make some synthetic skin, or not even synthetic, skin from stem cells. But skin's pretty simple, right? It's one layer of cells, two layer of cells, it's one kind of cell type, pretty easy. But why do you think in a test tube or in a beaker, why we can't make a heart or a liver or a kidney? It's huge. Specialized.
Needs other things from the body.
It needs other things from the body, and that's kind of getting on the right idea. As your own heart was developing, what was it next to? Your own heart was next to lungs and not too far from the liver. And there were certain products, gene products, proteins, that were in your body as an embryo that are no longer there. In other words, there are certain signals that were sent both temporally, time, at the right time, and there were right signals sent in the right space, spatially, that are necessary to form a heart, a liver, a kidney, a brain. We can't replicate that, at least not yet. It is going to be a very, very long time before we can replicate all of the chemical signals necessary to tell a heart cell to become a fully functioning heart, right? In fact, there's more than one cell, right? The heart is not just one cell, right? We know that organs like the heart are made up of epithelial, connective, muscle, and nervous. If I take a heart, there's some nervous tissue, there's some epithelial tissue, there's some connective tissue. So it's not a matter of just taking a few cells and saying, go get busy and grow and become something else. So that's where this perhaps is, the hope of creating a new spinal cord. Maybe, can we maybe repair some things? Maybe. Could we take cells, could we take a person who's had a heart attack, and could we give them some heart cells? And may those heart cells help fix the cardiac muscle damage? Maybe. Maybe. The hope is that we'll be able to take cells. Let's say this patient has diabetes, right? Type 1 diabetes. And their pancreas is no longer working and making insulin. Would it be possible for us to take cells and put them into his or her pancreas and maybe jumpstart the pancreas to making insulin? That's not too far-fetched, right? And that would be a kind of cloning situation. In fact, we don't need to take human embryos anymore to do this. And that's really the big news, and that's why you no longer hear about it in the news as much. Scientists are no longer forced to take embryos. Some would argue they need to, but they really don't need to. During that moratorium by Bush, we figured out other ways to get stem cells. We now know that in your own body there are stem cells. And these embryological cells are very similar. You can take cells from your skin. And down in the lower levels of your skin, there are cells there that are stem cell, like these embryological cells. And we've now, in the Petri dish, we used to think that once a skin cell was a skin cell, it was a skin cell, right? We couldn't make it do something else. They've actually taken skin cells, added the right chemicals and hormones, and reverted that skin cell back to becoming muscle. Actually, reverting it back to a dedifferentiated state and then forwarding it to become muscle. So the idea that a tissue once formed is stuck there may be too simplistic. So it might be that rather than taking cells from an embryo to implant in this diabetic patient, we could take skin cells from this very patient, trick that patient's skin cells back to a dedifferentiated state, trick them now forward to becoming pancreatic insulin-producing cells, and use his or her own cells to inject into her body to fix the diabetes. That's where stem cell research is going. So it won't be necessary, I don't believe, in 10 or 20 years to ever have this controversy over human embryos, because we have learned that potentially we can get these stem cells from not embryos but from adults. There's also a lot of talk right now about cord blood, right? Cord blood also has these stem cells in it, and parents have the option of freezing the cord blood. Why? Why do you freeze the cord blood? Maybe someday we'll figure out what to do with it, right? But we really don't have that many choices with it right now, but it is a really cool idea to have some of these stem cells set aside for you in the future should we ever have the technology to really understand what's going on. So regardless of all the hype that we hear about, embryological stem cells have not been really used to cure or treat disease. Everything we're hearing about so far, these stem cells are from adults. So bone marrow transplants, we've been doing bone marrow transplants now for decades. That's taking stem cells from your hip, right, from one patient and putting them into another patient. We're taking cells from one patient, putting them into another cell, and now into another patient, and now those cells start making the blood cells for the patient. So we've been kind of playing this game for a while, and now there are some treatments where there has been some regeneration of diseases, and this is a story about adult stem cells. So basically, I think the future is good for stem cell research. This is not going to go away, but we're going to find that almost everything we're doing will not require the destruction of an embryo, but instead can be taken from adult cells. So any thoughts on stem cells? I think it's an important conversation. I think we all need to be educated on this topic. Any thoughts on stem cells? Pro, negative? Some people have really heated ideas about this. So hopefully we don't have to ask this question anytime soon, right? How much human DNA is required for human status? Clearly we can move genes around from one organism to the other. Isn't that kind of creepy looking? But hopefully we don't ever have to be talking about transgenic organisms where we transfer entire genes from one organism to the next and get funky looking dogs like this. Okay, so let's start with the big picture. From the very first day we talked about the atom to atom hierarchy. We talked about atoms and molecules and macromolecules and organelles. That's kind of where we've been, right? Up through the cell layer. Now we're finally at tissues. So we're there in lab, we're now there in lecture. So let's go through tissues. What are tissues? Well, they are groups of cells that have a similar function. And you know there are 4 groups or types of tissues: epithelial, connective, muscle, and nervous. That's not news to you. What is news to you is what layers they are derived from. So, start at the bottom. Nervous tissue, where did I tell you it came from? The brain and the spinal cord came from the ectoderm. Where did your muscle tissue come from? Mesoderm. Where do the connective tissues come from? Mesoderm. Now, what about the epithelial tissues? All of them. If you hear me say epidermis, where is it? It's coming from the ectolayer. If you hear me saying mesothelium, it's coming from the mesoderm. If you hear me saying endothelium, it's epithelial tissues coming from the endoderm. So as you hear about those different theliums, the epithelium, the mesothelium, and the endothelium, you're being told right there what layer is coming from in the early embryo. So let's start with something you know the most about. You know a fair amount about epithelial tissues. Where do you find them? Right? They're covering our body. They're lining our organs. They're coating the inside of our cavities. And our glands are made up largely of epithelial tissue. They are tightly packed cells. You know that. And there's very little space in between those cells. That is, there's very little or no extracellular matrix. The cells are pretty much touching each other. There's no space for stuff outside of the cells. And there are no blood vessels penetrating through an epithelium. What else is true? Well, their cells. We know that their cells have polarity. Remember, one part of their cell is always glued down to the basement membrane, or is glued down to cells making a stack of cells. Another side of the cell is open to the environment, right? It's at the surface of your body, or maybe it's looking inside a tube. So that means that epithelial tissues have a polarity. They know from their north, from their south, if you will. The term used to describe their free surface, this is a new term, apical. The apical surface is the free surface. It's the top surface. Then the basal surface, the basal layer, is the layer that's on the bottom of the epithelial tissue that's usually glued down to the basement membrane. And the cells are tightly connected one to another through what is called intercellular junctions. Intercellular. What does inter mean? Between, right? So between the cells, there's something that holds them tightly together. So we know this. On the right-hand side, 3 different shapes of epithelial cells. We've got squamous, squishy, squashy, flat cells. You've got cuboidal cells, little cubes or squares. And then you've got tall rectangular prisms or columnar-shaped cells. Epithelia are described by shape and by layers. In the middle, if there's a single layer of cells, then it's simple. If there's 2 or more, it is stratified. Remember, too, that if it's a stratified epithelium, you always look at the shape of the cells at the outside layer or at the apical layer. And here, those cells are flat, so this would be stratified squamous. The epithelium is always going to be sitting down on a layer called the basement membrane. And the cells that are facing the basement membrane would be considered the basal surface of the cells. And again, the ones at the top are considered the apical. So apical and basal, basement membrane, these are terms that you've seen. Now, these cells are attached down to a basement membrane. Okay, down to a basement membrane. So I was thinking of that being glued down to a basement membrane. And what's below that basement membrane? Always, always, always some kind of connective tissue. Always, always. And epithelial tissues are avascular, no blood vessels. So what does that tell us? Where do these cells get their nutrients? Where do epithelial cells get their nutrients? If there's no blood flow, there's no oxygen and glucose being brought to these cells directly. So they must get their nutrients from the outside or from the connective tissues down below. Through what process? From higher to lower concentration, through diffusion. So now you begin to appreciate why we had to talk about diffusion so much, because that is the way by which many of our cells are getting their nutrients. So we need to appreciate the basic idea of diffusion. While there's no blood vessels, there are a lot of nerve endings. That is, epithelial tissues are highly innervated. The ability for you to touch, right? Feel pain and smooth and rough and pressure and vibration, all of those sensations that come through our skin, those nerve endings are certainly in our epithelial tissues, some of them, and we have hair follicles and we have receptors that come up into the epithelium. But most of that nervous tissue is down deeper in the dermis. Now, most of the dust in your vacuum cleaner is you, so your outer layers of skin are constantly regenerating. They are being scratched off. As you put on your clothing, there are cells being rubbed off. That means that they're being replaced. So we could say that your epithelial tissues are highly mitotic, right? Lots and lots of mitosis going on, lots of regeneration going on. And deep in your skin levels are where those adult stem cells lay. Now, these cells are not screaming, I'm a stem cell. So when you take cells from your skin, some of them are stem cells. They don't really go into action until you cut yourself. And when you cut yourself, suddenly your skin regenerates quite nicely. What allows your skin to regenerate are those adult stem cells that are sitting there, and they're sitting at the basal layer. So they're sitting at the deepest layer of your epidermis. Now, what do these cells do? We know that epithelial tissues protect us, right? We're protected from water loss. We're protected from virus and bacteria. We know that they create a selective permeability. When you hear the word permeability, up until now, I described that with cell membranes, right? That it's the cell membrane that allows certain molecules into and out of a cell. But your skin also creates, epithelial layers create a selective permeability. Some things can get into or out of or across these tissues. And what's brand new is that epithelial tissues, many of them produce secretions. Secretions are made by two different types of glands, endocrine glands and exocrine glands. What does endo mean? Within, crine, to secrete. So endocrine glands are going to secrete something within the body, whereas exocrine glands are going to secrete something out of the body, like sweat, tears. Already mentioned nerve endings, right? There are some nerve endings definitely in your epithelial tissues. So let's take a look at this picture. Simplified picture. Now, what do we have here? We've got one layer of cells, so it's a simple epithelium. It's sitting on a basement membrane, and below it there is connective tissue. So we've seen that story before. But this particular cartoon has something extra labeled that is brand new, and that is that your basement membrane is not a single layer, but it's actually a double layer. Okay. So your basement membrane is actually two layers. There's a basal lamina and a reticular lamina. Now, whenever you hear that word lamina, lamella, lamina, laminated countertop, right? Something very thin, like a layer. So lamina this, lamina that. Two layers. The top layer is the basal lamina. The bottom layer is the reticular lamina. And they're labeled for you over here on the side. Basically, do you agree so far that I've told you that epithelial sit on the basement membrane, and the basement membrane below it, there's connective tissue? Mm-hmm. Basically, the basement membrane is two things. The basal lamina, made by the basal cells of the epithelium, meet the reticular lamina, made by the connective tissues below. So the epithelial tissues and the connective tissues are each making a part of this thing called the basement membrane. But this is where the two layers are connected or touching each other. So the basement membrane, two layers. Now, under the light microscope, you only see one layer. You would never be able to see these two layers, but they are separate layers, two parts to the basement membrane. I don't know why I put this slide back in there. So we know this well, right? We know this well. Being a little sluggish. Now, we know that epithelial tissues are tightly packed. That means that they're being held together by something that glues them together. And these collectively are called the tight junctions. There are 4 types of tight junctions, and I'll list them each for you here. Number 1, tight junctions. Okay, this is one of the 4 types of cell junctions. Tight junctions. Tight junctions. What you have over here on the cartoon. This is one cell. This is one cell. Here's another cell. Those two cells are touching each other. They're actually being knitted together. And at the top, this almost looks like Charmin, right? Some sort of toilet paper being quilted together. that top layer is the tight junction. It's typically found up toward the top at the apical surface. Okay? So they're going all the way around the epithelial cells up by the apical surface. There's also adhering junctions. They are also forming circles around the entire cell, but they're a little bit lower. They're down closer to the basal side. Okay? Now, what these tight junctions and adhering junctions are going to do is make sure that molecules can't squeeze between the cell layers. Right? They're creating a barrier. So molecules can't squeeze through between the cells. They must be welcomed into and go through the cells, which creates a greater selectivity, right? Creates a greater process of controlling all of this. Now, the third and fourth type are desmosomes. Desmosomes are really cool. I think of desmosomes as rivets. Where on a pair of jeans do you find rivets? At the pockets, places where there is more stress. So tissues that are under greater stress will have desmosomes. They are like little buttons, little snaps, and each cell provides half of the snap button process complex. So you're going to find these in the heart, right? The heart is always beating. That's a lot of stress. You'll find desmosomes in greater numbers in the vagina or in the anus, places where there's stretching and a lot of stress going on those tissues. Finally, there are gap junctions. Gap junctions are like little pores, little connectors between. So what we have here in this picture is here's a cell on the right and here's a cell on the left. So we got two cells, and these cells are connected by these gap junctions. These are like little channels allowing for nutrients to travel from one cell laterally to the next cell. So we got things holding cells together, but we also have ways for cells to communicate one with another. So all four of those cell junctions are shown in this one picture. So if you're going to look at a picture, this is the one I want you to know. So take a moment, put a little asterisk on this picture, because this is the one you will see again. All four of these cell types are all these junctions are found. So here's tight junctions, adhering junctions, desmosomes, and gap junctions. Don't worry about the Latin names that are associated with them. Just know them by those four English names. I also like this picture because it shows that the basement membrane is really two layers. The upper layer, again, was the basal lamina, and the lower layer was the reticular lamina. So this picture does a couple things, right? It shows both layers of the basement membrane, and it shows all four of these intercellular junctions in one picture. Remember, epithelial cells, tightly packed, right? Tightly packed. Very little extracellular matrix. Now, you also know that epithelial tissues are based upon their layers. We've got that. We've got the simple, and we have the stratified. Where you have a simple epithelium is where there's less stress. Right? Wherever you have a simple epithelium, you don't have a lot of protection needed. So where do you find simple epithelium? Are they anywhere near the outside of the body? No, they're deep inside the body. Where are they? Things like the alveoli, the little air sacs in your lungs. Things like the lining of your intestines, absorbing nutrients. Things like lining the tubules of your kidneys for filtration. You're not going to find simple epithelium exposed to the outside world because it's only one cell thick. But you know this to be true. What do you have inside your nose, inside your nasal passages? What kind of epithelium is inside your nose? Do you know? Inside your nose is the same thing that goes down your trachea. Pseudostratified. Is pseudostratified simple or stratified? It's really a simple epithelium. And this is why it's so easy for your nose to bleed in the wintertime. Right? If you've got a dry nose and you just scratch the inside of your nose and it starts to bleed, why? It's not stratified. It's not multiple layers. It's a single layer of cells. They're kind of jumbled up, but it's a single layer of cells. So it makes that area much more prone to bleeding, right, to destroying that layer. So it's much easier for your nasal passages to get raw and inflamed because of that. Now, then if there's stratified epithelium, again, where do we find these stratified epithelia? This is your skin, your epidermis, as well as, and that should say 2 or more, okay, 2 or more, not one of more, but 2 or more layers. Where else do you find stratified epithelium? Say it again. The mouth, yeah, so inside, right? So what kind is inside the mouth? Stratified, squamous, non-keratinized. Down the esophagus, same thing. Anus, vagina, same thing. Stratified, squamous, non-keratinized. And what would you look for if you were looking at that under the microscope? Many layers, but at the top the cells are still alive. They're squamous, they're alive, and we can still see a nucleus. Good. And then there's that pseudostratified one, right? It's that funky one that we looked at, and sorry, I'm having slow responsiveness on the iPad. So pseudostratified, it looks stratified at first, but at closer examination, if you could truly pull apart those cells, you would see that they all are reaching down and touching the basement membrane. And what do we always see on pseudostratified? Cilia, right? So in the respiratory tract. So that includes up in your nasal passages. So we've got our 3 different shapes. Now, squamous, cuboidal, columnar. Now there's one more that you don't have to recognize under the microscope, but I want you to know about, and that is transitional. There was a question in the pre-lab about this as well. So transitional is a special kind of epithelium found lining the inside of your bladder. And as your bladder fills and empties, it transitions. As your bladder fills, it stretches like a balloon and is very, very thin. And when you void and the urine leaves and the bladder now shrinks, that outer layer kind of jumbles up. So it transitions and changes its shape all the time. That's where it gets its name. You're not responsible for recognizing it under the microscope, but I do want you to know about transitional epithelium. So I've got a quick journey through these tissues. You've seen these, or ones just like it. So sit back, enjoy the tour, and confirm your understanding. Now you know that you're going to be quizzed on histology in really kind of 3 ways. 1, pictures, right? And I'll always have nice pictures like this or what you saw in lab presentation last week. Number 2, you need to know a little bit about them. So there may be some word questions about histology. And then thirdly, during the lab practical, you'll actually be going to stations with microscopes and the tissue will already be there, the slide. And so you'll need to be able to recognize it under the microscope. So you need to be thinking about histology in a few different ways. This is one of those ways where I'm showing you nice pictures. Martini textbook's great for this. It really is. So in there you've got page after page of histology and things are labeled. It's nice. You've got your PAL resource. You've got your atlas. You've got plenty of places to go to look for and practice this. In this particular view, this is actually pointing to just the upper layer. The simple squamous cells are right here, this top layer. You can see a single layer of thin cells. This is actually coming from, this is a blastula. Okay, you don't need to know that, but that's the blastula. This is that hollow ball of cells, right, that fluid-filled area. And there's a single layer of cells around the top of that thing that are simple squamous. But where are you going to see them? You're going to see them in our tissues down in the alveoli, in the little air sacs of the lung, and you're going to see them lining blood vessels, and you're going to see the simple squamous as your serous membranes. The visceral and the parietal layers of the pleura, pericardium, and peritoneum are in fact simple squamous epithelium. And that's where you see these other words. I've already said this. So simple squamous epithelium could be endothelium, lining your blood vessels, could be mesothelium, your serous membranes. Second one. Simple cuboidal. Now in lab, you only saw this as a circular arrangement of cells. And that's a good place to start. But imagine now that that tube that was going in and out of the board in lab has now been opened up long axis. So now you're looking at, let me draw a basement membrane on here for you. You're looking at a tube that's going here and here. And this tube is going left to right. And you're seeing just a little bit of this layer. And it's one layer of cells, and they're all rather cuboidal looking. So you're cutting into that tube long ways. So that is simple cuboidal. And as it says, kidney tubules are the place to think about. That's the primo place to find simple cuboidal. It's present in other places as well. Keep it simple. You only need to know one. Next one, simple columnar. Now, there's two different types of simple columnar, with and without cilia. Okay? Non-ciliated and ciliated. What did we look at in lab? When we looked at simple columnar, that was from the intestine. And there, did it have cilia or not? Yes. It did not. It had microvilli. Okay, it was non-ciliated. Cilia and microvilli never coexist on the same cell. It's either they're there, one or the other, or neither, but never both. So what we saw was non-ciliated. We saw, lining the intestine, microvilli, right? Microvilli. And they were there to increase the surface area for absorption of nutrients. There are other examples in the body where simple columnar do have cilia, and this would be along the uterus and along the bronchioles, the bigger tubes of airways going down into your lung. There would be some cilia. The one, though, that you've seen and dream about, right, is this one. So this is simple columnar. You're looking, the white space in between is where the food substances will be passing through your intestines. So the food is traveling, well, I shouldn't say both ways, right? Food is traveling in one direction, okay? And let me get you your bearings on this. Basement membrane. Basement membrane. You're looking at a tube, aren't you? Okay. And this tube is surrounded by this simple squamous. And you can't see it. Remember, you cannot see microvilli, but I do see sort of a little fuzzy border along this. So that fuzzy border is called the brush border, and that's the evidence that there is microvilli there. And what do we see visiting? What other cell type are these big white things? Goblet cells, right? You want your gut nice and moist. So those are the mucus-producing cells. Now, are there simple columnar epithelia with cilia? Absolutely. As this says, these are along the fallopian tube. You can see them in this little picture up here. And if you look closely, you can see individual cilia on this micrograph. So it does exist. Then there are the stratified squamous epithelia. You know there's two types. We've already talked about all these things. There's keratinized and non-keratinized. Which one is this? Just don't look at try not to look at the words. Just look at the picture. Stratified, squamous, non-keratinized. What do you notice? Here's the basement membrane, right? Down here, you see the color change. And all these epithelial layers, but at the surface, I still see nuclei. So there's only two kinds of stratified epithelium you need to know, right? They're both stratified squamous. One's keratinized, one's non-keratinized. So here I definitely see epithelia, flat cells at the top, and I definitely see nuclei. So again, they're alive and well, oral cavity, down the throat, esophagus, vagina, anus. Those are the places you'll find stratified squamous. It is protective. Versus, now look up at the screen. Try not to look at the words. Get your eyes to work for you. The blue, you haven't seen blue before. The blue is a different stain, but the blue is the connective tissue down below. So everything below the blue is connective tissue. Everything above it, many, many layers of epithelial cells. At the surface, very flaky and dry, dead, no nuclei. You can even see those little light spaces. Those light spaces are an artifact. As this tissue was put on the microscope, those layers are beginning to kind of pull apart from each other. So that's the dust in your vacuum cleaner as those things begin to slough off at the surface. About halfway up, what do you see happening? I see a distinct difference, don't you? You see a distinct difference right about here? Everything below that is living. Everything above that is dead. Now, are there stratified cuboidal epithelia in the body? Yes. Do you need to recognize them? No. So this right here is just more of a gee whiz, let's look. But there are stratified cuboidal cells, and they're found in your salivary glands, for example. And there are also stratified columnar, 2 layers of columnar cells, and these are found in the male urethra and in some other glands. I'm not asking you to know these. Okay, these are just, oh, by the way, they do exist. These are the same ones we crossed off in lab, right? When we crossed off some of those circles, these are the ones that we crossed off. Pseudostratified, this one's important though, right? We've seen this one. We see cilia on the surface, individual little structures. I can see big, white, empty spaces. Those are the goblet cells. Now, mucus is kind of like fat. We don't see it there, right? But just know that it's being made in those cells. The basement membrane's down here. So you can see below that, it's all connective tissue, and above that, what appears to be multiple layers of cells, but it's not. Transitional, again, not to recognize it, but to, I do want you to know about this one. Again, this is found in your bladder, and it relaxes. In this particular view, it's quite relaxed. So your basement membrane's here, and it looks like a bunch of layers of cells, doesn't it? And the reason I don't have you learn this one is it kind of looks a lot like pseudostratified, and your eyes aren't quite trained to see the difference very easily. So we won't learn this one under the microscope, but do know that it's in the bladder. And those are the different epithelial tissues in your body. Now, the other thing that you'll find made up of epithelial tissues are your glands. So I want to tell you a little bit about glands. 2 basic flavors, endo and exocrine glands. Here's the difference. Endocrine glands, you have an entire endocrine system. What is that system doing? It's a bunch of glands that make hormones, that release those hormones within the bloodstream. In order to do that, they just simply kind of open up the cell and drop the stuff into the bloodstream. So there are no ducts. They lack ducts, tubes, and they drop their products directly into either the bloodstream or into the special fluid between your cells. So when you hear about the endocrine system, those are the endocrine glands. Think thyroid. Think pituitary glands. But many of the glands in your body are exocrine. That means they're releasing something out of the body. In order to release those products out of the body, there must be a tube, a duct. And so exocrine glands have a duct, a tube, and they carry those products to the surface of the body. Think sweat glands. Right? There's a little tube that carries that sweat to the surface of your skin. This even includes your tear glands. That makes sense. Tears are outside. But this also includes your digestive glands. At first, you might not think of your stomach as being exocrine, but it is. When your stomach makes acid, that acid then mixes with the food and then travels through your digestive tract and leaves your body. Exo. So everything going from mouth to anus down the middle of your body is actually outside of your body. Does that make sense to you? When you eat food, that piece of food traveling through your gut system and then eventually defecated, that is, from an anatomical standpoint, outside of your body. It's a tube traveling through the middle, but it's literally connected to the outside world. Whatever is absorbed from that food into your bloodstream now is within your body. But as long as that kernel of corn stays and becomes part of your feces, that is outside. Exocrine. So here, what I'm going to be talking about here are exocrine glands. For the next 5 or 6 minutes, it's all about exocrine glands. This is a cartoon representation of an exocrine gland. This is the entire gland or part of the gland on the left. If you zoom in to one little tiny section right here and zoom in, this is what you would see. It's a duct, right? We see a tube, we see a duct, and it's like a little cul-de-sac. It's like a little neighborhood, right? Cells at the end of the street. And all of those cells are producing stuff. So these are epithelial cells at the end, and these little secretory vesicles, right? These cells are making proteins. Those proteins are being synthesized by ribosomes, packaged by Golgi, packaged in little vesicles, and those little vesicles are exocytosed out into this cul-de-sac, into this duct. And then all those products go out, don't they? They go out the duct. So maybe to the surface of the skin. Maybe this is your tear gland. Maybe this is your pancreas making gut enzymes, gut juice. To me, this looks like a fancy neighborhood. You know, you're flying and you look down and you see this neighborhood, right, with all these little cul-de-sacs, this neighborhood. And all the houses are accessed from this main street that goes into this neighborhood, right, the duct. So that's kind of my visual, what I see here. Now, it turns out that glands can be classified in many different ways. One, by their structure. Another way of saying that, their morphology, their morph, their shape. So we won't spend a lot of time on this. Some glands are simple, some glands are compound. I'm not going to distinguish between those two. I do, however, want to take a moment to talk about their method of secretion. Glands have three different ways of doing their business. They can work by merocrine, holocrine, or apocrine. And I've got a slide for each of those to distinguish those three. And then glands can either put out a very watery substance called serous, very serous fluids. They can put out a very thick fluid like a mucus, or that gland can be mixed. Okay, just kind of a combination. So let's take a look. First of all, I told you I'm not going to worry about this. So you could really put a big old X through this, but I want you to see some glands are very simple, some are very complex. In other words, the simplest of glands just have a little, indentation, and others look like that fancy neighborhood. Right? Not to distinguish them, just see simple to complex glands. What I do want to focus on, though, is their way of doing business. The first one is merocrine. Now, merocrine, this is how your salivary glands work. This is how your tear glands work. This is how your gut juices are produced by the pancreas and by the stomach. We're looking at, again, one of those little cul-de-sacs of cells. These are the epithelial cells on the bottom. They release their product into the duct. The duct eventually carries the product to the surface of the skin or to the surface of the body somewhere. That's merocrine. Hold on to that idea. Look at the second one, and this is holocrine. Now, there's something very distinct about this. This on the bottom represents a little group of cells. The cells are dividing, but as they release their products, these cells actually release themselves. So the cells are part of the product. They're not just releasing a fluid, they're releasing junky cells. This is the kind of secretions made by your facial sweat glands. This is a big reason for acne, right? So as the cells are releasing their substances, they're getting clogged up by this cellular debris. Now, the way I remember this, the whole cell is being released, holocrine, right, as in W-H-O-L-E, whole thing, versus, and the cell dies, right? The cell is dying and being released and kind of this junk, versus, going back, in merocrine, the cell stays merry, if you will. The cell is still very much alive, right? So the whole cell is not dying, the cell is staying intact. So that's how I just put that one together in my head. So here the cell is intact, it's not dying, it's merry, it's releasing its product, it's very watery, versus holocrine, where it's much chunkier in its secretions and the cell is dying in the process. Again, the oily glands of your face are released this way. Finally, there's apocrine. Apocrine is like an intermediate version. It's half merocrine, half holocrine. This is how breast milk is released, as an example. Apo here refers to apical. So here's the lumen, right? We're looking in that tube. The cells are releasing their products, but notice it's like the cell is pinching off. So it's releasing a little bit of the cell. The cell's not dying, but the cell is releasing some of itself. So it's a little bit chunky, but not clogging chunky. And this is how, again, breast milk is released. So it's the apocrine is referring to the apical side of the cell is released. The open side is pinched off and released. So I do want you to know merocrine, holocrine, and apocrine. And then the secretion can be serous-y or mucus-y. Serous-y, watery. Sweat, very watery-like. Tears, very watery. Digestive juices, you don't know it, but your pancreas is making a lot of juices. They're very watery. Or mucus, very thick. And then mixed glands, you'll appreciate this. Your salivary glands are considered mixed glands. When you're about to eat, your salivary glands make very watery secretions, right? You're about to digest food, they become very watery. But when you're scared, you become very dry. And those secretions become very thick, don't they? Or you wake up, right, and in the morning, and a little thicker. So you know your salivary glands can put out watery kind of stuff, or they can put out thick stuff, so we call them mixed glands. We'll talk about that more when we get to the autonomic nervous system, why sometimes you put out a thick secretion and why sometimes it's more watery. I've only got about 5 minutes, but I want to at least get through the introduction to the connective tissues. Then that doesn't leave that much, to be honest, because you're not going to become experts in muscle tissue or nervous tissue. I want to introduce them to you. You will be responsible for what muscle tissue looks like under the microscope, but I'm going to give you very clear images, and next week in lab, you'll be looking at muscle tissue under the microscope. So this is all going to come together nicely. So how are connective tissues different than epithelial tissues? This is important from last week's lab, right? We know it's a very broad group, right? We get everything blood, fat, tendon, ligament, right? Bone. All of those things are made of connective tissues, and we know that they are very diverse. They are doing all sorts of things for us, not just connecting us together, but many other things as well. And this includes, like I said, the dermis, ligaments, tendons, blood, fat, cartilage. They all come from the mesoderm. Right? That's an important fact. I've already mentioned that. They all are derived from the mesoderm. And again, the functions are very, very broad. You could argue that fat and bones both protect us. You could argue that cartilage gives a structural framework, that ligaments bind and hold things together. What do ligaments do? They bind bone to bone. Fat also, not only does it protect us, but it also stores, right? We're storing energy. Bone stores calcium. So we've got a lot of other functions going on here. We've got transportation going on. The blood is transporting not only glucose, but oxygen and waste products, hormones. And your blood is also a big part of your immune system when you think about your white blood cells that are protecting. So there's lots of different functions of connective tissues. Here's a table that may help you put this all together. So at the very top, it says connective tissues, and I know it says common origin mesenchyme. Just cross it off and put mesoderm because that's the word we've used. Right, so all of your connective tissues are derived from the mesoderm. And there's 3 basic groups of connective tissues. Fluid. Well, there's only one in this family, right? Only one fluid connective tissue. You know what that is. That's blood. There are 2 that are considered supporting. You know these as well. Cartilage and bone. Under bone, this week in lab, you'll learn a little bit more about spongy bone. What we looked at under the microscope last week was compact bone. Under cartilage, there's 3 different types. Last week we only looked at hyaline, but I'll mention throughout the course the other 2, fibro and elastic cartilage. Those are the connective types, or the supporting types. And then the rest of them are thrown in this big old garbage pile called connective tissue proper. This includes loose connective tissues. Now we looked under the microscope and we saw one called areolar loose connective tissue. But this is also where fat belongs. So fat is also a loose connective tissue. And then last week we also looked at dense, regular connective tissue. Now there's 2 other types. We're not going to look at them this semester. So you get the idea, right? We're just trying to categorize these different connective tissues. And I'm going to finish with this slide today, so hang in there. What is it that makes all of these things, blood, fat, connective, all part of the connective tissue family? What are their characteristics? Loosely packed cells, right? We got that. Now those cells could be fibroblasts or adipocytes or chondrocytes or osteocytes, right? Or blood cells. Those cells are surrounded by a ground substance that's a non-living stuff, liquid, solid, or semi-solid. And mixed in that ground substance, there will be some protein fibers. And again, I mentioned this in lab. I wrote this on the board. There's collagen fibers that give tissue strength. There's elastic fibers that give tissue stretch, elasticity. And there are reticular fibers, as the name suggests, create a network, a scaffolding, a basic building block on which other things are built. Collectively, the ground substance and the protein fibers are called the extracellular matrix. Okay, ECM. So what I'll do is on Wednesday, I'll just walk you through these. You've already seen blood and bone and cartilage. It's not going to be much of a stretch. I'll introduce muscle tissue to you as well as nervous tissue, one slide. And then we'll jump over and talk about skin, which actually is very straightforward. And we'll definitely, I think, get through that comfortably on Wednesday. And that will all be part of the exam next Monday. Come chat with me if there's any concerns. If anyone listening, when they started the course, got into mastering using a temporary code, certainly that temporary code by now has expired. It's time to get that flipped over, so let me know. There's a couple of students I see in the roster who are still using the temporary access. That will have expired by now. So please don't forget that there's quite a few assignments on mastering, getting ready for this exam. So go ahead and get those started so there's no emergencies this weekend.